this video, we will perform a genetic association analysis of cardiomyopathy in the St. Jude Life cohort. Before beginning the analysis, we'll set up a subcohort of survivors. To do so, we will open the Filter tab of the portal, and we will filter survivors for those that have been exposed to either anthracycline chemotherapy or heart radiation. So we'll add a new filter and search for anthracyclines. We'll choose cumulative anthracyclines. And we'll set the values of this variable to be greater than 0. And then we'll add a OR term and search for the variable heart radiation. And we'll set the values of this variable to be greater than 0. So now all those survivors in this subcohort have been exposed to either anthracyclines or heart radiation. Then we'll set up our regression analysis by going to the Charts tab, and we'll click on the Regression Analysis button, and we'll set up a logistic regression. The outcome variable for this analysis will be cardiomyopathy. And the grades for the outcome variable will be grades 3 to 5. So we'll edit this grade cutoff to be 3. And then we'll add independent variables. The first variable we'll add will be sex. Then we will also add an independent variable for age of diagnosis. We can slightly modify the bins of this variable. Set the last bin start to be 15. We'll also control for age at last assessment. We'll also modify the last bin of this variable to be 55. Then we'll also control for exposure to anthracyclines. And we'll edit the bins of this variable as well. We'll set bins of varying sizes. First will be 0, then 100, then 250. We'll change the boundary inclusion so that the first bin is less than or equal to 0. And we'll just rename this bin label to be not exposed. And then we'll also control for exposure to heart radiation. We'll also modify the bins of this variable, varying bin sizes. So this is 0, 1,000, 3,000. And then also change the boundary inclusion so that this is less than or equal to 0. And we'll rename this to be not exposed. Okay, and lastly, we will make this analysis a genetic association analysis by adding a genetic variable. So in this case, we'll select variance in a locus. And to specify the locus, we'll enter a variant this variant has previously been shown to be associated with cardiomyopathy in African ancestry. So after entering the RSID of the variant, the coordinate of the variant is retrieved. And then we'll also restrict the analysis to African ancestry. We'll keep all the other settings for the genetic variable as default. And we'll submit. So now this genetic variable has been added to the regression analysis. And now we can run the analysis. So when a genetic variable is used in the regression analysis, the results are shown in a genome browser view. And the only variant shown in this view is the same variant that we inputted into the analysis. And as you can see, the variant is plotted on this y-axis according to its p-value that was computed in the analysis. When we click on the variant, we can see its complete regression results that were computed in the analysis. We can also zoom out the view to analyze the neighboring variants in the same analysis. So if we zoom out twofold, we still don't see neighboring variants, but we can zoom out even more 50-fold. And here we can see several neighboring variants that neighbor our variant of interest. And all of these variants were analyzed in the same analysis. However, they were analyzed in separate regression models so that they were not treated as covariates within the same model. We can even zoom out further by specifying the gene of the gene model that this variant is found in. So this variant is found within the MAGI3 gene. So we can specify MAGI3 within the search box. And now all variants within MAGI3 will be analyzed in the regression analysis. So you can see here the 300 plus variants within MAGI3 have been analyzed in the regression analysis, and they have all been plotted according to their p-values that were computed in the analysis. And when we click on our variant of interest, you can see that all variants within the region have been color-coded according to their linkage disequilibrium values relative to the selected variant.